Dear students, Assalamu alaikum. This is lecture 16 of the blood physiology. In the last lecture we studied the humoral immunity, and today we'll study the cell-mediated immunity, the immune tolerance, immunization, and immune reactions. To understand the cell-mediated immunity, we need to know the types of various T cells and their functions. T cells are classified into three major groups: helper T cells, cytotoxic T cells, and suppressor T cells. They can also be classified according to the CD classification. CD means cluster of differentiation. The leukocytes on their surface possess certain antigenic molecules, which are identified by monoclonal antibodies, and they're numbered CD1, CD2, CD3. The CD4 positive cells are the helper T cells. They can bind with MHC class II molecules and activate B lymphocytes to form the antibodies. The CD8 positive T cells are the immature T cells. They can bind with MHC class I molecule and they differentiate into cytotoxic T cells. Now let's study the helper T cells. The helper T cells are by far the most numerous of the T cells, constituting about 75% of all of them. They serve as the major regulator of all immune reactions. They do this by forming a series of protein mediators called lymphokines that act on other cells of the immune system and also on the bone marrow cells. The important lymphokines secreted by the T helper cells are interleukin 2, 3, 4, 5, and 6, granulocyte monocyte colony stimulating factor, interferon gamma. The lymphokines have a specific regulatory function. In the absence of the lymphokines from the T helper cells, the remainder of the immune system is almost paralyzed. In fact, these are the T helper cells that are inactivated or destroyed by the human immunodeficiency virus, HIV, thus leading to the debilitating and lethal effects of acquired immunodeficiency syndrome. The other function of helper T cells is the stimulation of growth and proliferation of both cytotoxic T cells and suppressor T cells. The lymphokine interleukin 2 has an especially strong stimulatory effect in causing growth and proliferation of both cytotoxic and suppressor T cells. The helper T cell also cause stimulation of B cell growth and differentiation. Almost all the interleukins participate in the B cell response, but especially interleukins 4, 5, and 6 and they have such potent effects on the B cells that they have been called as the B cell stimulating factors or B cell growth factors. The helper T cells cause the activation of the macrophage system. Firstly, they slow or stop the migration of the macrophages after they have been chemotactically attracted into the inflamed tissue, thus causing great accumulation of macrophages. And secondly, they activate the macrophages to cause far more efficient phagocytosis, allowing them to attack and destroy increasing number of invading bacteria or other tissue destroying agents. This diagram shows the regulation of the immune system, emphasizing a pivotal role of the T helper cells. Now let's study the cytotoxic T cells. The cytotoxic T cell is a direct attack cell that is capable of killing microorganisms and sometimes even some of the body's own cells. The receptor proteins on the surfaces of the cytotoxic cells cause them to bind tightly to those organisms or cells. After binding, the cytotoxic T cell secretes hole-forming proteins called perforins, which can cause holes in the membrane of the attack cell. After making holes in the membrane of the attack cell, the cytotoxic T cell releases cytotoxic substances directly into the attack cell. Immediately afterwards, the attack cell becomes greatly swollen and it usually dissolves shortly thereafter. The cytotoxic T cells, they can kill tissue cells infected by viruses, and they also play an important role in destroying, number one, the cancer cells and also the heart transplant cells. The diagram shows the direct destruction of an invading cell by different sensitized lymphocytes. The suppressor T cells. The suppressor T cells can suppress the functions of both cytotoxic and helper T cells, and therefore the suppressor cells are classified along with the helper T cells as regulatory T cells. The suppressor T cells play an important role in limiting the ability of the immune system to attack a person's own body tissues. And this phenomena is known as immune tolerance. We studied this phenomena when we were studying the pre-processing of the lymphocytes in the thymus. The process of acquired immunity would destroy the individual's own body if a person became immune to his or her own tissues. The immune mechanism normally recognizes a person's own tissues as being distinctive from bacteria or viruses. The tolerance results from clonal selection during pre-processing in the thymus and bone marrow. It's the failure of the tolerance mechanism that causes the autoimmune diseases. The destruction of body's own tissues releases self-antigens that cause acquired immunity in the form of either activated T cells or antibodies. Immunization has been used for many years to produce acquired immunity against specific diseases. Remember that immunization is a form of acquired immunity. A person can be immunized by injecting dead organisms that are no longer capable of causing disease, but that still have some of their chemical antigens. This type of immunization is used to protect against typhoid fever, whooping cough, diphtheria. Immunity can also be achieved against toxins that have been treated with chemicals so that their toxic nature has been destroyed, even though their antigens are still intact. This type of immunization is used to protect against tetanus and botulism. And finally, a person can be immunized by being infected with organisms that have been attenuated. This type of immunization is used to protect against poliomyelitis, measles, yellow fever, and smallpox. Now let's study passive immunity. Passive immunity is one of the subtypes of the acquired immunity. All the acquired immunity that we have discussed so far has been the active immunity, which means the person's own body develops either antibodies or activated T cells in response to invasion of the body by a foreign antigen. Passive immunity is a temporary immunity that can be achieved without any antigen. This is done by administration of preformed antibodies or activated T cells. The antibodies or activated T cells are obtained from the blood that has been actively immunized against the antigen. Antibodies can last in the body of the recipient for 2 to 3 weeks, while the activated T cells last for few weeks if transfused from another person, and from few hours to few days if transfused from an animal. Such transfusion of antibodies or T lymphocytes to confer immunity is called the passive immunity. This table shows the comparison of active immunity versus the passive immunity. Now let's study the disorders of the immune reactions. The disorders of immune reactions can be grouped as one of these four reactions: hypersensitivity reactions, or they may be rejection phenomena, autoimmune diseases, or the immune deficiency diseases. The hypersensitivity reactions are of four types. The type 1 reactions are of immediate onset, occurring within minutes, and the antibodies involved are the IgE antibodies. The effector cells are mast cells and the eosinophils. Important diseases because of the type 1 reactions include, first of all, anaphylaxis. Anaphylaxis is a serious allergic reaction that is rapid in onset and may cause death within minutes. Anaphylaxis can occur because of the insect stings, medication, and the food allergies. Other diseases in which the type 1 reactions are the cause include the atopic diseases like rhinitis, asthma, and urticaria. Rhinitis is the inflammation of the nasal mucosa resulting into nasal discharge. Urticaria are the palpable skin lesions The type 2 reactions are of intermediate in onset. That is, they take hours for their manifestation. The antibodies involved are the IgG and IgM. The effector cells are the neutrophils and the macrophages. The important diseases in which the type 2 reactions form the basis include the autoimmune hemolytic anemias, myasthenia gravis, This concludes our today's lecture.